3.2 Carbohydrates. Carbohydrates are almost universally used as an immediate energy source in living things, but they also play structural roles in a variety of organisms. See Fig 3.1. The majority of carbohydrates have a carbon to hydrogen to oxygen ratio of 1 to 2 to 1. The term carbohydrate includes single sugar molecules and also chains of sugars. The chain length varies from a few sugars to hundreds of sugars. The long chains are thus polymers. The monomers of carbohydrates are monosaccharides. Writing energy. Monosaccharides, monos, which means singles, and saccharide, which means sugar, consisting of only a single sugar molecule, are called simple sugars. A simple sugar can have a carbon backbone of 3 to 7 carbons. The molecular formula for a simple sugar is some multiple of CH2O, suggesting that every carbon atom is bonded to an negative H and an negative OH. This is not strictly correct, as you can see by examining the structural formula for glucose, Fig 3.6. Still, sugars do have many hydroxyl groups, and this polar functional group makes them soluble in water. Glucose, with 6 carbon atoms, is a hexose, hex, which means 6, and has a molecular formula of C6H12O6. Despite the fact that glucose has several isomers, such as fructose and galactose, we usually think of C6H12O6 as glucose. This signifies that glucose has a special place in the chemistry of organisms. This simple sugar is the major source of cellular fuel for all living things. Glucose is transported in the blood of animals, and it is a molecule that is broken down in nearly all types of organisms during cellular respiration, with the resulting buildup of ATP molecules. Ribose and deoxyribose, with 5 carbon atoms, are pentases, pent, which means 5, of significance because they are found respectively in the nucleic acids RNA and DNA. RNA and DNA are discussed later in the chapter. Disaccharides, varied uses. A disaccharide contains 2 monosaccharides that have joined during a dehydration reaction. Figure 3.7 shows how the disaccharide maltose, an ingredient used in brewing, arises when 2 glucose molecules bond together. Note the position of the bond that results when the negative OH groups participating in the reaction project below the ring. When our hydrolytic digestive juices break this bond, the result is 2 glucose molecules. Sucrose, the structure shown at right, is another disaccharide of special interest because it is sugar we use at home to sweeten our food. Sucrose is also the form in which sugar is transported in plants. We acquire sucrose from plants such as sugar cane and sugar beets. You may also have heard of lactose, a disaccharide found in milk. Lactose is glucose combined with galactose. Individuals that are lactose intolerant cannot break this disaccharide down and have subsequent medical problems. To prevent problems, they can buy foods in which lactose has been broken down in its subunits.
Polysaccharides, energy storage molecules. Polysaccharides are polymers of monosaccharides. Some types of polysaccharides function as short-term energy storage molecules. When an organism requires energy, the polysaccharide is broken down to release sugar molecules. The helical shape of the polysaccharides in Figure 3.8 exposes the sugar linkages to the hydrolytic enzymes that can break them down. Plants store glucose as starch. The cells of a potato contain granules where the starch resides during winter until energy is needed for growth in the spring. Notice in Figure 3.8a that starch exists in 2 forms. One form, amylose, is non-branched, and the other, amylopectin, is branched. When a polysaccharide is branched, there is no main carbon chain because new chains occur at regular intervals, always at the 6th carbon of the monomer. Animals store glucose as glycogen. In our bodies and those of the other vertebrates, liver cells contain granules where glycogen is stored until needed. The storage and release of glucose from liver cells is under the control of hormones. After we eat, the release of the hormone insulin from the pancreas promotes the storage of glucose as glycogen. Notice in Figure 3.8b that glycogen is even more branched than starch. Polysaccharides serve as storage molecules because they are not as soluble in water and are much larger than a sugar. Therefore, polysaccharides cannot easily pass through the plasma membrane as a sheet-like structure that encloses cells. Polysaccharides, structural molecules. Structural polysaccharides include cellulose in plants, chitin in animals and fungi, and peptidoglycan in bacteria. See Fig 3.1. In all three, monomers are joined by the type of bond shown in cellulose in Figure 3.9. The cellulose monomer is simply glucose, but in chitin, the monomer has an attached amino group. The structure of the peptidoglycan is even more complex because each monomer also has an amino acid chain. Cellulose is the most abundant carbohydrate and, indeed, the most abundant organic molecule on Earth. Over 100 billion tons of cellulose is produced by plants each year. Wood, a cellulose plant product, is used for construction, and cotton is used for cloth. Microorganisms, but not animals, are able to digest the bond between glucose monomers in cellulose. The protozoans in the gut of termites allows termites to digest woods. In cows and other ruminants, microorganisms break down the cellulose in a special pouch before the cud is returned to the mouth for more chewing and re-swallowing. In rabbits, microorganisms digest cellulose in a pouch where it is packaged into pellets. In order to make use of the nutrient pellets, rabbits have to re-swallow them as soon as they pass out of the anus. For animals such as humans that have no means of digesting cellulose, cellulose is a dietary fiber which maintains regularity of elimination. Chitin, GK chiton tunic, is found in fungal cell walls and is in the exoskeletons of crabs and related animals such as lobsters, scorpions, and insects. Chitin, like cellulose, cannot be digested by animals. However, humans have found many other good uses for chitin. Seeds are coated with chitin, and this protects them from attack by soil fungi, because chitin also has antibacterial and antiviral properties. It is processed and used in medicine as a wound dressing and suture material. Chitin is even useful during the production of cosmetics and various foods.
Figure 3.1 is shown. In Figure 3.1, it shows a cactus, a crab, and a bacteria, which all contain carbohydrates. They contain cellulose, chitin, and peptidoglycan. Explain why humans cannot utilize the glucose in cellulose as a nutrient source. Humans have no means of digesting cellulose. Compare and contrast the structure and function of cellulose with chitin. Chitin and cellulose cannot be digested by animals. Chitin is found in the fungal cell walls and the exoskeletons of crabs and related animals, while cellulose is found in plants.